But this is one of those. Yeah.
Just like
You leave it in for like a minute.
Yeah.
Just yes.
When that one's done calibrating, you close it out, you pull it back. Then you can rinse it and put it back to the tank. Don't turn the solenoid on. You let that sit for a minute. And then when you basically do like you leave it in the tank.
yeah, that's
He's fine. He likes it.
Check.
Is it sharing screen there?
keep up with analytics on this.
But I certainly can tell it was probably regular. I don't have to contact him about, you really should be looking at,
We should do what AFS is doing now. Just not ask.
you know, the question, you'll probably. All right.
All right. Well, thank you, everyone, all my crayfish folks out there, for joining us for Reggie Turner's public exit seminar as part of the requirements for his degree. So you know, we've had a few of these in the last couple of weeks. So excited that we get to Reggie today. And as usual, as we like to do for our labs, you know, the traditional roast, right? Who is Reggie Turner, right? You all had a chance to meet Reggie. This is actually a great picture that kind of describes, you know, Reggie, you know, like being a crayfish. And you know, we typically try to really take it way back, and knowing that Reggie's mom is here. But as I was looking through Reggie's history here and his time, I decided that probably this was not the approach, although Reggie's super happy to see this, right? Christmas time. But just general background on Reggie. This is the Reggie that we know out there in the field. He earned a bachelor's degree in biology from Georgia Gwinnett College. And at his time at Georgia Gwinnett, he assisted as a field and lab research tech for one of the faculty out there. And also, actually, I found out, as I'm kind of going back to the files, he even spent a month-long summer studying abroad in China and Japan. So when I saw that, it got me to think this is something that we probably don't do a lot. I went back to Reggie's application when Reggie applied to join the program, because, you know, years happen, and then you're like, wow, like, what was it about Reggie? And one of the neat things that I found about Reggie, I pulled one of his reference letters. Just going to read really briefly what one of his references said, and actually clicked a lot today, and hopefully as you see the presentation, it'll even make more sense to you. One of his reference letters said, I threw a lot at Reggie, and Reggie seemed to pick up all these things very quickly. Not only that, but he developed new techniques himself, and at the end of it, he was teaching me how to run statistical programs. So eventually, he suggested a program called Beast. And Reggie proceeded to teach his advisor about Beast. And he said, I should have read this right before we started with this. But one of the neat things about Reggie is that clearly, yes, he did a lot of that. He was able to take in a lot and then really advance what we were trying to do in the lab, which is kind of neat, because if you think back to Reggie when he was an undergrad, he first wanted to go to med school, right? Then he, you know, switched paths and maybe wanted to go to vet school, work as a vet technician for several years from high school till even after his undergraduate degree. But that experience with that one particular professor that allowed him to be a field and lab technician really clicked and changed his perspective as to what he wanted to do after. And I'm glad that it did, because clearly Reggie brought a lot to our lab. A lot of the traditional things, presented his research a lot got awards to go to conferences like the Society for Freshwater Science as an endowed award member for a travel fellowship. Even participated in the National Diversity in STEM conference, or SAGNAS, also with a travel scholarship. And for several years, he was a Society for Freshwater Science Emerge Fellow, which is an NSF fellowship, so pretty productive. So Reggie did a lot in terms of using that platform for presenting his research. Not only that, one thing that I think Reggie, we will all agree, at least in our lab, has been great at, is really taking the forefront in presenting about his work in an outreach standpoint. In the one project that we work the most now, in a large collaboration between North Carolina, South Carolina, and Georgia, Reggie has really been a good example as to what we're trying to learn for crayfish communities in Georgia, particularly the Ogeechee River. And I think our colleagues have really got to appreciate what Reggie has brought to the table, like you see him here presenting in one of our project meetings. But it hasn't only been in that space. Reggie has also done this at Carver Elementary in Richmond Hill with a lot of kids, and you can see, look at all the future crayfish ecologists. So clearly, I'm proud of everything that Reggie has done, which brings me back, just very briefly, the last thing I'll do, to read from Reggie's statement of purpose as he joined Georgia Southern. Reggie wrote, "In the future, I'd like to pursue a PhD in ecology. I hope you still do at some point, which I discovered a passion for in my undergraduate years. I love learning about ecology more in the classroom and in the field than any other class." Reggie then said, "My professor warned me that much of my time will be spent off campus, occasionally knee or hip deep in stream with over 30 pounds of fishing gear. But he also cleverly forgot to mention the massive mosquitoes that likely travel in swarms. But even with all the negative aspects, I still love every minute of being out there and in the lab doing the genetic work, doing the lab work, doing the field work. And if given a chance to do it again, I will take it without any hesitation whatsoever. And I would love to do that sort of work to the point where being paid to do it would just be an afterthought." So proud of you, Reggie. Clearly, you've gone a long way in teaching us how to go from this to that. So with that said, I'll pass it on to Reggie. Excited to hear about your work. I lost the title already, but I'm sure you'll tell us a little bit more.
Well, thanks for coming. Appreciate you all making some time to come here today. So before we start, I kind of wanted to thank all the people that really helped out with this project. So most importantly, all the students, the lab alumni, and friends who've helped with the research, as well as my committee who's all here today for all the help and guidance that they've had from me over the years. And well, the 20s also to my family who supported me through all this. So thank you to everybody who's contributed to this. All right, so just starting out with some basics. So why crayfish as a model? So crayfish actually are very important in our streams for multiple reasons. The top three are just kind of here. So when we think about crayfish as prey species, a lot of our game fish like bass and like Arlis and Trunket species, they'll eat them, and also their purpose of like moving nutrients up to the environment is very important as a prey species, as well as bringing those nutrients from the sediment. So they break down leaf litter, and in doing so, they also kind of help to move the nutrient from from dead plants and also animals in some cases up the food chain. So that action of moving nutrients is pretty vital for how crayfish are actually contributing to this process. And they also will form a very unknown kind of thing for a lot of people. Most of them realize that they actually burrows. So actually they are found underground. And when they're actually underground, they're also aerating the soil and also kind of providing homes for a lot of either frog and also salamanders that also can make homes in these little holes. And in doing so, they're also not just for themselves providing these homes, but they're also kind of contributing to all the different interactions, both in the water and outside of the water. So crayfish in that way are actually pretty important to the environment. And also when we think about like trends of biodiversity across the Southeast, when we talk about like the state of Georgia in particular, it's not terribly in peril in terms of our watersheds. Tennessee's actually got way more of our perils attack. So, but even in Georgia, we still have quite a few watersheds that do have a lot of these imperiled species, including with crayfish. So you can kind of see there's up in the north near the mountains, we do have more in general of our tax species. But in the south, we still have an appreciable number of watersheds that do have imperiled taxa for crayfish. So there is a good reason to kind of focus a lot of our efforts in the state here in the southeast. So when we think about, like, from a legal standpoint, where a lot of our conservation efforts are currently, so we have for about 60% of crayfish taxa are threatened to some degree. So they're either critically imperiled, or vulnerable to some degree in the state of Georgia. So unfortunately, that's more than half, so that's not great. And about 7% of our species, or at least 5 that we know of, are non-native to the state. That includes Procambarus carki. So if you've ever been to a crawfish boil, the big red crayfish that you eat there, those are the ones that are invasive to the state. They're the biggest offenders. There's a few others, but that one in particular is one of the worst ones. And we have one species that's considered data deficient by Georgia DNR. So it's actually listed as well in the State Wildlife Action Plan for last year, Procambarus petersi. You see it there. That's actually one of the few that's in the state that is a watershed endemic as well. It's only found in the Ogeechee River. And it was actually recently described in the 1980s, and we don't have a lot of habitat distribution data for this species in particular. So not only is it rare, we don't know much about where it is. So it's good kind of focus species to focus on at least. So in terms of legal protections, there's less than 30% of the species have any sort of laws on the books to protect them currently. But currently, in terms of like things that DNR has done recently, about 38%, almost 40% of species have been listed as species of greatest concern, is the term that they use in the Georgia State Wildlife Action Plan for last year. So the Georgia DNR is actually doing a good job of helping to get more of these taxa listed for protection of the state. All right. So why the Ogeechee? So as like a river to focus on, it's actually pretty common in terms of it being a black water river here. So if you're unfamiliar, black water is just referring to the color of like the tannins in the water. So we think about, for example, clear water streams up north in the mountains being different from the black water streams we have here in the coastal plain. It also kind of reflects the differences in flow regimes here. So when you think about up in the mountains, there's faster moving water. The waters tend to be more channelized. The floodplain connectivity is a lot less. So, and as that water moves towards coastal plain, the rivers tend to like belly out a bit more. So there's more floodplain connectivity. And also the water has more time to leach tannins out of these leaves and turn to much more darker color, leading these black water streams. As we move from the Ogeechee, which is headwaters up in the Piedmont, actually, so about 6% of the basin's in the Piedmont, the rest is actually down below the fall line. As you move through the fall line, you actually get more of this kind of like these black water or like wetlandy kind of areas. And as we do, we also see diverse changes in the habitat. So different tree cover in terms of going from more of our kind of like hardwoods to more of like pine dominant, and also more cypress trees in a lot of these wetland areas. And also, something I kind of want to touch on is nothing that's really specific to the Ogeechee itself is the fact that there are no major impoundments for this river, which it's actually the longest unimpounded river in the state. It runs about 300 miles, so about 450 kilometers from its headwaters near Crawfordville all the way to basically kind of where Gilly Island is kind of where the mouth is. There are no dams along that entire stretch. So when it comes to it being a good model for a lot of these different species, and also our migratory fish, we can assume that the migration is unaffected along the coast. And also another thing I want to touch on is the fact that the flood pulse is a very critical part of this river system. So when you think about the flood pulse just being this period, like a hydro period of like increased flow in certain times of the year and then decreased flow for the rest of the year, and over a year and year after year, there's this almost like eccentric clock to it, kind of predictability in the change of flow regime throughout the year. And there's a lot of studies that have been done on crayfish in particular, having a greater kind of like reliance on this system of increased flow regime and kind of flooding these wetlands, providing a lot of almost like a nursery effect for a lot of our migratory fish that go spawn and fry, and also but for crayfish they also require this constant this yearly kind of cycle throughout the basin. So also I want to touch on some of the crayfish species that we have here in the basin. There are 18 known species according to the Georgia DNR, and through this study I actually found 11 of them. So there's a few more that as a lab we found in other places. So for example, like, Muncie, Howell, and Seminole, we found other parts of Georgia, but for the Ogeechee River in particular, I've only found 11 for what we've done so far. I just wanted to take a couple minutes to kind of think about putting basically the names. This is the same more that we saw earlier. So Canberra's laddermantis, also known as the variable crayfish, pretty common up north toward the Piedmont. Also have Lacuna Canberra's diogenes, which is the devil crayfish, Procamberus acutus, which is the white river crayfish. So this is the total amount of genera that we have, just the 3 that we caught in this study. But the basin has a total of 4. So Faxinellus is the only genus that I didn't find in this study that we should expect to find. Procamberus avena is actually another one of our burrowing species. You can kind of see it has big claws on its hands there. We also know the dollar crayfish, also Enopisternum, which is the black mottled crayfish, which tends to live. We found it typically in the main stem along the Ogeechee, as well as some of the tributaries in the upper and lower. Procamberus epicertus is the humpback crayfish. Procamberus lodesternum is the blackwater crayfish. Peter's eye, this is the one that is kind of like our poster child for this project. At least this is the Ogeechee crayfish, known only for being found here in the Ogeechee River Basin. Procamberus pubescens, which is the brush-nosed crayfish. Troglodytes, which is the eastern red swamp crayfish. So it's different from Clarkii, which is the true red swamp crayfish, native to the Mississippi River Basin. Troglodytes is our native kind of cousin to that species, pretty much found throughout Georgia and all the way up towards like the southern end of North Carolina. And then last but not least, we have Truculentus, which is the bog crayfish. This is our last of the burrowing species that we found. All right, so I kind of want to take some time to think about river network perspective. So how can we think about how diversity changes across the riverscape? So the first being longitudinally, so thinking about how communities change from the headwaters all the way to the mouth of the river. So basically thinking that ecoregions or that sort of view is what is primarily driving these changes in community composition, versus a lateral kind of perspective, which is kind of saying that the main stem might have its own unique community or groups of communities which are separate from the tributaries that surround it. And then last but not least, the watershed perspective, that's saying that each of these different hucks or watershed subbasins has its own unique communities, and the barriers between these subbasins that are dividing them are acting as kind of like barriers to migration or the species ranges between these communities. Right. And also kind of touch on community kind of structure, meta-community structure. So if you want to know what the meta-community is, it's really just like a set of local communities that are linked by the dispersal of interacting species. That's the most basic thing I kind of think of for this. And it's determined by 3 main points. So we have coherence, range boundary clumping, or range turnover. And it's just a flow chart. So you can kind of think, so if coherence is significantly negative, it's checkerboard. If it's non-significant as a test, it's random, so on and so forth. It's a very simple way of just at least characterizing the structure of meta-community based. And this is just kind of like, I kind of want to show you this so you can see what it looks like when you look at a site by species matrix, or if you look on a map of the ranges of overlapping. So the first one there is evenly spaced. So you can kind of see like a staircase pattern. And also each species kind of overlaps with one other species in this range. So that's one idea. Or the second that you see up there is the idea of Clementsian kind of meta-community structure, which is each community has its own species that are found in nowhere else. And there's no overlap in species in each of those communities, so on and so forth. So you can kind of see there are different kind of outcomes for each of these meta-community structures. So, and also separate from the idea of meta-communities are the ideas of communities of common profile. Works a little bit differently. So it's really just groups of these communities with common species occurrence probabilities that are more similar to each other within that group and other groups with different communities. This is more of like a regional focus rather than like being by dispersal. So we don't really care about dispersal for communities of common profile. It's really just which of these communities have similar probabilities of occurrence for each of these species. Think of it almost like a combination lock in a way. So each group has its own kind of combination. For example, like species A has like more like a 70–80%. Species B has like 52 to 15%, and so on and so forth, which might be different from some other group combination for that series. That makes sense. So our research questions are really just incorporating the fluvial structure of the watershed. So you remember the longitudinal, the latitudinal, the watershed, just incorporating the river network position of each of those points significantly improve our explanatory and predictive powers of the models that we create with this, and also how best can we group these assemblages in a geographic context to explain patterns of crayfish composition across the basin, as well as how can we characterize the structure of meta-communities after we're done with all of this. So this just kind of just puts these in hypothesis form. So fluvial structure is a significant predictor of crayfish assemblage composition, as well as there are distinct clusters of these communities of common profile for crayfish throughout the basin, as well as there are in the structure is best described as Clementsian, just off the bat. That's the working hypothesis that we're going for meta communities. And there's an easy test for this afterwards, so we'll test it out here and there. So just going over the methods. So we use typically for our passive traps, we either use our ProMars or the minnow traps. There's a ProMar here that we have deployed at the site. And we also have our minnow trap. So you see that black object there, which is actually one of our minnow traps. And the traps are left overnight. Every time we set traps out, they were left overnight, collected the following morning. And we always have 10 traps each of our sites. And for our active settling protocol, we did dip netting and tick seining, as well as hand collections anytime that was warranted. So the next picture you see here is of a dip netting setup, of a tick seining setup. So typically we have at least 2 people who are in the net, and on a third person actually kicking, kind of using using their boots like a shovel, kind of like scooping basically everything on the benthos into the net. Typically for crayfish stuff, we usually also kind of like wrap a chain at the bottom of the net because crayfish are different from fish, so they aren't found inside the water column; they're always at the bottom. So we tend to make some modifications to our protocol there. And our transects were typically 30 times the wetted width. So we always measure the wetted width and we multiply that times 30 to get the length of our sampling kind of like regions for each of those. We used 100-meter mins and 500-meter maxes, and burrows were always excavated whenever we found them. So that's me a picture of me digging a burrow up there. And sometimes we actually got crayfish. So typically, if we found females and berry, we just identified what they were and just kind of put them back. Anytime we had males, we usually pick those guys up. And we always identified our stuff in the field typically, so any species we found, we always identified them, and then just noted how many there were and also some habitat characteristics typically. So when it came to our sites, the Ogeechee has 27 what we call hut tents. So if I'm familiar with the hut system, these are just kind of like almost like Russian Matryoshka dolls. There's so many that fit into each of these categories. So we have, for example, like 4 hut gates, and within those hut gates, we actually have 27 hut tents. Each of those has a name, and we chose 2 sites per hut tent was our goal. So we have 1 smaller order and 1 larger order stream per each of these hut tents, and they're all selected by connectivity to the main stem. And we also used some previous lab data from previous RUs and RAMs that were here previously for a total of 37 sites. And so for our variables, we have 4 different kind of groups. The first being land covers, so that will be like developed versus agricultural versus forest cover and so on and so forth. And versus hydrological, which would be like our landscape hydrological variables, like distance to the mouth, distance to the main stem, upstream channel length, those sorts of things. Versus geographical cover, which would be more like slope, ecoregion type, geological type. So like if we have gneiss or granite or sandstone, whatever is our major geological type underneath the ground there. And last but not least, biological. So that would be like what species are actually found in each of these sites. So I don't want to I kind of want to at least set this up for what we're talking about later. So these are the different stages of like statistical analysis that we did to get to where we ended up. So the first thing I did was kind of like this gradient forest analysis. So familiar with random forest analysis, it's very similar, except that it also performs kind of like this species accumulation plot. So I'll show this in a bit as well. So basically, I was really just using that to figure out which of these variables in a machine learning kind of way are the most important in describing how assemblages are changing across the basin. So I took, the best kind of predictors from that and also kind of threw them into what we call like a variance inflation factor test, which is really just testing for multicollinearity. So making sure that they are kind of like fighting for explained variation. So after we got those, we put those variables, we separated them into 2 categories. We had our categorical variables and continuous, and we tested them in different ways. So we had the threshold indicator tax analysis, also known as Titan, which for our continuous predictors, and we also use indicator species analysis for our categorical variables. And the variables that significantly explain variation across space from those tests were placed into stage 3, which is model generation. So we used 2 different models. We had multivariate generalized linear modeling. We also had something else called Bayesian model called hierarchical modeling of species communities, which works. So the first one, the multivariate GLM, is a frequentist approach versus a Bayesian approach for the second one. It could also be, I'm sorry, another way that that was a little bit different is we also had random spatial effects for the Bayesian that we couldn't use for the multivariate GLM. So we could add either each of the Euclidean points in space that they were on a map, or we could also include a distance matrix of like the distance between every other site along the fluvial network. So we use use that to figure out if using the distance between sites as the fish swims explains more variation than the sites as the crow flies. So we use the output from the HMSC, and that actually gives us basically the predicted species occurrence probabilities, which are how likely we are to find each species at each site. So it gives you a number between like 0 and 1, so 0 being 0%, 1 being 100%, how likely each species is to be at each site. And having that data, we also threw that into the last step, which was our cluster analysis. So we had the probability data, and we fed into 2 different independent clustering methods to see if we get the same clusters out of it. Basically, kind of like a stability test to make sure that the clusters are as robust as they should be. So the first being what we call a tandem approach, which was a principal component analysis followed up by partitioning around medoids. If you're unfamiliar with medoids, I'll go over those just a sec. And also the other one being the hierarchical clustering of principal components, or HCPC for short, which uses k-means clustering set of medoids. And again, I'll go over more of that in a second. So just for now, I kind of wanted to just, before we get too deep into stats, go into where we found stuff across the basin. So for all the species that we see here, that's every species that we found in the study. There was some that kind of stuck out. So for example, Peter's eye, the light green that you see up at the top there, those were only found in the upper Ogeechee. We didn't find them anywhere else, which kind of matches up, which is a little bit different from what previous studies have found, for that species, which was much further south and also in the Canoochee as well. So we didn't find it in the Canoochee, we didn't find the lower Ogeechee, pretty much only in the upper Ogeechee, which we found a few new sites, but also most of them were relatively where we were expecting to find them. We also found Litosternum, which was mostly in the Canoochee, which is that the one to the left, the black group to the left there. We also found Epicertus on the other side of that divide in the lower and coastal Ogeechee tributaries. Troglodytes was kind of cosmopolitan. We found it all over in every one of the subbasins. We found pubescens, mostly in the upper lower Ogeechee main stem, as well as Enobosternum, also in the main stem and also in the Canoochee as well. And Latimanus we only really found pretty much in the upper Ogeechee, and as well as in one site near Millen at the Ogeechee for main stem. So another thing I want to mention here, when we think about like the natural history and like the study of crayfish kind of like collections historically, some of these species, as far as my results go, we found some range expansions, or at least some places where we did not know that they were there before, basically. So, for example, Enobosternum, we found along the main stem pretty far south, and we also found them extending almost close to Crawfordville, which is pretty far north. Historically, Enobosternum is much more common in the Oconee and the Hoopie. There's very few records of it being in the Ogeechee. So the fact that we found so many records for them in the Ogeechee is a pretty good sign that there's definitely a larger population than we knew previously for those species. And also we found pubescens in a lot of different places than we had known prehistorically as well. And that just kind of goes to show like there's a lot of these species that are probably cryptically in places that we just don't know. There's no one who's really doing this research. That's part of the reason why I was so interested in finding out where some of these species. So just going over gradient force analysis. So we have, for example, 3 of the consistently top results that came up every time. So wetland cover, total channel length, as well as forest cover. Those are our top results. I ran this kind of like 10, 15 times just to make sure that every test gave the same result, and the top 3 came out with just those 3 every time. And also top 3 species in the machine learning way of being the best, being predicted the best, were Peter's eye, Litosternum, and Enobosternum. And this is what I was talking about. So one of the differences that you can kind of have between gradient force and random force is that you can kind of see along those gradients where there are changeover points, where places where along that range there is a great increase in like a presence or decrease. is kind of like the absolute values, what we're capturing here. It does not tell you if the direction of the change. So it doesn't tell you if that's increasing or if it's decreasing, but it does tell you that something's happening. Right, so we took the ones that survived from those tests and we used the continuous variables we put into the tight analysis. So this one here is wet and cover. So we can see, so the blue colors for this are decreasers. So as the gradient increases, the environmental gradient increases, these decrease as that increases. So for example, P. petersii tends to drop off at around 23% wetland cover versus troglodytes, which after 30%, it increases. So we see more of them. We expect to see more of them after as that increases versus our TUCL or upper channel length, which is our Engelstrom and pubescence. Those two tend to increase as the stream size increases. Versus for categoricals, we had multiple of our categories that were significant. So we think about, for example, hugates, the litosternum and pubescence. Those were the significant groups with litosternum, with the Canucci and the lower and upper Okichi. And also for some of our other ones, like in my ecoregions had positive results, or had significant results. Watershed zone. So I actually cut the watershed into 3 parts just to get an idea for if there were certain species that were associated with different bands within this watershed. That had significant results, as well as site type, which was our main stem versus tributary sites. So we saw pubescence and Engelsternum as having a positive kind of significant relationship with our main stem sites. So after we kind of threw those into the models, we took wetland cover, upstream channel length, as well as hugates to form a model. And so what I did was I actually tested a whole bunch of the combinations of different variables until I found one that actually were all 3 of the terms were independently significant when accounting for the other 2, as well as all 3 having a low multicollinearity. They're not struggling to explain variation on their own. So also it gives, when we run an MVGLM, it also gives us coefficients. So you can actually get an idea for which of these are significantly have a significant either positive or negative correlations with our values. So we had 2 positive results here. So we had troglodytes, which had a positive significant result with wetland cover, which is expected. And also pubescence having a strong positive significant relationship with total upstream channel length or stream size, basically. So the other modeling method we had was HMSC. So why did we do it? So for example, the other one that I mentioned was MVGLM, which is the frequentist approach. And one of the biggest issues with our frequentist approach is that complete separation is a big deal. So we found that some of these hugates are actually what we call perfect predictors, meaning that they always predict where a certain species is because they're never found anywhere else in any other categories. So they actually the model can actually assign standard error to a lot of these, and so it just gives a massive range, saying it's somewhere in here somewhere. So that's why you see these ballooning standard errors for some of the hugates, but we don't see that for wetland covering TUCL. There's enough error in that so they can properly get built in standard error range for those. But for many of the species, the results for hugates were unreliable due to the standard error being so large. But Bayesian statistics works differently. So we can actually use as priors, and it walks around that distribution across 10,000 or so samples to build up enough error to be able to calculate standard error. So we can kind of get around that idea. And also another big thing is with this particular HMSC method, it also allows us to provide spatial random effects. So kind of like I mentioned earlier, we can tell the model to use the XY coordinates for each of the sites. We can also tell it to use a distance matrix of every distance between every other site along the river network to kind of say how best does this distance explain where stuff is basically in the basin. So this is kind of like a graphical interpretation of what I'm kind of mentioning. So if we took every site and we planted a 10-mile buffer around each site, all the sites that are within that buffer, this program is kind of saying, or trying to test, are each of the bubbles, each of the dots within this bubble more similar than everything else outside of this bubble, basically. As opposed to, this is the Euclidean model versus the Fluvian model, which is saying, okay, so now we know where stuff is along the river network. Can we make predictions on basically how big that range is from this site to this site? Does that predict, the differences in community structure across the basin? So in that case, we built 2 different models to test this. We built the Fluvian model that uses this versus the Euclidean model, which uses our XY coordinates. So kind of an idea for how much each variable accounts for variation within each of the different models we had. For example, we saw some big differences. So we had for litosternum, which is the middle bar in each of those, there was a big difference in the random spatial effect. So Euclidean versus fluvial. We saw that the fluvial system actually had more variation explained by the distances between sites versus in the Euclidean model, which is just the difference of distances as a crow flies within a certain range. Versus, for example, Troglodytes, which actually had most of its explained variation being kind of eaten up by wetland cover. So overwhelmingly, we can kind of like get an idea of where stuff is based on is there enough wetland cover for this species, for example. And for both of these models, we found that puck gates actually, as an average, tend to explain the most variation in these, and also that the random spatial effect actually explain the least typically for most of our species. But also, it is interesting to note that they had relatively equal contributions. So if you look at the means up top, we had the ranges were typically between 27 and like 36%, so close to 30% for each of the terms. So they're all kind of like pitching in equal amounts rather than having one that actually like overwhelms everything else as a term. And this is just kind of like the coefficient plot that we saw for MVGLM, except that it kind of calculates it a little bit differently. So we have more significant results in this one. So we have, for example, for total upstream channel length, we saw that both pubescens and enobosternum had positive results. So our positive results are in red, negatives are in blue. So we also saw that we had positive significant results for those two species. And we also had a negative result for Epicertus. So that was something else kind of keep in the back of your head. We'll talk about more in a sec. Also, I want to mention that middle bar again is, so the bar, the middle bar there, litosternum. We have positive, so significant results in the Euclidean model, but we have no significant results in the fluvial model. So it kind of ties back to what we saw earlier. So that random spatial effect is actually pulling a lot of the explained variation out of the other terms and leaving it so that the other terms are insignificant when it comes to explaining what stuff is. Another thing that we'll kind of think about is now we have gates, which is different. So before we didn't have gates for the coefficients, but now we do because the calculus looks differently. We can see that, for example, pubescens has a positive result with with the upper Geechee, and also for wetland cover in the top right corner there, that red dot, the red square with black dot, that's Troglodytes having a positive relationship with the wetland cover. So we actually did build models for this, and we did figure out that, for example, Euclidean model and the fluvial model, actually, the WAICs were very similar to AICs. The models were actually more parsimonious. They actually did a better job of explaining, even relative to the number of terms we threw at it, it actually did better. And also between the Euclidean and the fluvial models, we did find that the fluvial model did have lower WAICs for each species, except for just one, which is Pinguicernum. That was the only one. And we also found that Troglodytes and pubescens actually have the highest WAIC, and we also have the Epicertus and Peter's eye having the lowest WAIC. So the model is actually doing a better job for or that's more parsimonious for the species. And I actually took all three of those models, and I actually got some of the summary statistics. So the AUC, or the area under technically the receiver operating curve, and also the TSS, which is the true scale statistic. Those things are just ways of kind of looking at how often did it get stuff right, and also like how specifically was it correct. So we found that Latimanus was actually the worst one. Whoops, sorry. Latimanus, the one at the bottom, performed the worst out of each of the species. And also that orange bar at each of those is our fluvial model, and it tended to work the best out of the three. So out of our MVGLM, the HMSC Euclidean model, which is like the XY coordinates, versus using the as the fish swims, the as the fish swims model actually performed best for most of our species. And also what I did was I took those that sites by species matrix of probabilities of occurrence, and I actually performed PCA with those, and it actually gave me, and that color coordinated. So PC1 is red, PC2 is green, and PC3 is blue. And we did find that there were actually kind of distinct clusters, which is kind of cool to see. So you can kind of see like we have a collection of red dots up top, you know, blue kind of like group of dots in the Canucci. We also have kind of like this green kind of set of dots kind of like wrapping around the main stem. And the main stem itself is that kind of like light orange and kind of greenish color. So and you can see that both in the geographic space and in the PC space, it's even more apparent. So that kind of leads us to our next kind of question of like, how can we best characterize the differences in these kind of groups of communities? So something I kind of built was like, I took that same PC model and then I added a third axis, the PC3 actually. And we did find that, for example, those blue and green dots there on the 2D model, we found that those were kind of superimposed on each other. But actually, when we take that third axis out, we see that those do separate along that third axis, which is pretty cool to see. So we have 4 major groups. We have kind of like an upper Ogeechee, which is our red dots there. We have a Canoochee, which is our blue dots. We have that lower coastal tributary group, which is the green dots there, the darker green. And we have the main stem corridor group, which is that light brown and that light green color that you see there. And we also kind of did the same thing just in 2 dimensions, just to get an idea for a PC space, how they split out. And we used these are the tandem results. We have the PCA, which is principal component analysis, plus I took those PCA coordinates and threw them into the partitioning around medoids. So if you're familiar with a medoid is, it's a spatial medium. So it's all it is. So it is always a representative site within your data set as opposed to a centroid, which is kind of like whichever point, or you can actually make up a point that minimizes what they call the sum of squares distance, which is kind of like the distance between that center and every other point in the data set. But it doesn't necessarily have to be a point for a centroid, but for a medoid, it always is. So kind of 2 different ways of thinking about where these are in space. I also performed a silhouette analysis, and we found that the upper Ogeechee and the main stem corridor groups were the most distinct and had the best separation out of all the 4, versus the 2 on the left, which are our Canoochee and lower coastal Ogeechee groups, which are quite as distinct. They're a little more what they call fuzzy. They're much more adjacent to each other in the PP space and geographically. And so much more with this is just an NMDS of the presence absence of our species. So we found that, for example, Litosternum is actually doing the primary job of moving the Canoochee group out, versus, for example, like Peter's eye, which is pushing the upper Ogeechee a little bit away from the main stem group. And there's more overlap between those 2 than either is to the Canoochee group. There's actually 0 overlap in species between those 2 groups. But the giant green group, which is our lower coastal Ogeechee tributaries group, acts almost like a bridge. So it actually has almost all the species found in each of those categories together. And so just for my sanity, I actually kind of performed kind of like a sensitivity analysis of the PCAs as well as just the raw probability data alone. So just to get an idea for if the PCA there does a better job of separating things out in space. So we found that the size of the PCA groups was like 7, 12, 8, and 10. But if you go to the size for the raw probability, which is different, the site memberships are different between the 2 clustering groups. And for example, you can see 7 and 12, and then in raw probabilities, it's 6 and 13. So in the raw probability group, 1 got stolen into the lower coastal Ogeechee group. And also you kind of see, I don't want to get too deep into this, but you can kind of see, for example, for the diameter and the average distance, those numbers are smaller for the PCA group. So there are what they call more distinct. There's better separation. And so that's kind of like all I kind of want you to focus on there. And also when we talk about just like the differences between those 2 methods, we found that the overall width was better with the PCA group versus the raw probability group. The objective, or kind of like how hard the model has to work to kind of organize stuff, was actually much worse with the raw probability data, and actually had an easier time doing it with the PCA data. And the negative silhouettes, which is kind of like our misclassification number, that was actually, we had more misclassifications with the raw data set than we did with the PCA data. So moving on to the other method, this is the same PCX as we saw before. So we had PC1, PC2, and this is just the height of a dendrogram showing how similar each of these are with each other. So we find that, for example, the Canoochee and the lower coastal Ogeechee groups are closer to each other in species probabilities than either is to, for example, upper Ogeechee and the main stem corridor. So this is the same dendrogram we saw earlier, just kind of smashed down to just like 1 axis there. And this is all of the sites that you see here are also kind of labeled over there. So you kind of see in space, they do cluster. So we have, for example, all of our Ogeechee sites in the upper Ogeechee. All the Canoochee sites are in the Canoochee, and also the main stem sites are along the main stem of the Ogeechee, and the lower coastal sites are kind of wrapped around the main stem corridor. So when we talk about, like, for example, the relationship between these clusters, we do find that, for example, the lower coastal Geechee groups are closer to each other than, like how we mentioned before, but also that this group has like a much lower branch height, so there's more species overlap, and there's a greater kind of like there's actually a lesser difference between the species probabilities in each of these sites. So as an example, we have little sternum on the left hand there and trunk values on the right hand there. So the idea is that, you know, there is much more kind of overlap in these species ranges as opposed to the other group, which has much more differences in the probabilities of occurrence for these species. So for example, we have the Ogeechee crayfish Peter's eye and pubescens there. And if we were to just draw like a line in the sand between the upper Ogeechee sites and the main stem corridor, we do see that, for example, the Ogeechee crayfish doesn't cross that line, but for example, the pubescens or Burstone crayfish actually is found in both of those categories. And also just kind of like giving you a reference for where we're finding the Flatamatis, we typically found in our upper Ogeechee group, we tend to find Peter's eye, and also in the upper Ogeechee, we tend to find pubescens in the main stem and the upper Ogeechee. We also tend to find Lusternum mainly in the main stem corridor, but also occasionally in the tributaries in the upper Ogeechee. Little Lusternum was typically found in the Canoochee or yellow sites there. We also tend to find Epicertus in the lower coastal Ogeechee group, the blue groups that we see there, and we find Pogadius, the eastern red small crayfish, typically in our coastal wetlands, which is in the south there. And these also provide, we can kind of get an idea for how this method works, is we can get an idea for each of those clusters, how much work is each species doing in defining where these clusters are, and also where how much work that each of those axes are in defining or pushing these clusters away. So for example, in the Canoochee, the little sternum or the blackwater crayfish has a very high positive association with this group, and also the other three negative. So it's actually decided by both of those. It's defined by the presence or like increased probability of one, and also the negative probabilities of the others at the same time. You can also get an idea for, I just kind of want to show, so we have an idea for all three of the first three PC dimensions were critical in defining where these were, and only those three. So it actually kind of justified why using that third axis rather than just going off just two rows. And so I also performed indication species analysis based on the new categories, and we found that it has, we did find good indicators for each of those categories. Except for the last one, C-Letamatis, which was the only one that didn't have a positive kind of like a significant relationship with any of them. And so I also performed some sensitivity stuff. So I performed a pairwise urbanova on the new groups, and we found that for both the presence absence and the Hillinger transform abundance data, it was significant for each of those are significantly different between the groups. So just coming back to the meta community stuff, we did find that the coherence was negative, significantly negative. The boundary or range tolerance was non-significant positive, and the boundary clumping was also positive, but not significant. And that leads us to believe, because coherence was significantly negative, we just stopped there. So that is a checkerboard pattern. That's it. But also I did a bit of digging, and also checkerboard is kind of like a weird thing when you think about communities. It's harder to tell between checkerboard and random. There's a bigger problem there. And that just kind of means that it's hard. This actually works by kind of compressing all the variation along one axis and trying to organize things along that axis. And basically, if you have either one of those top things, it's really saying the model is struggling to put everything on one axis. It's very likely that we're dealing with multiple environmental axes there. All right. So this is kind of just like a representation of the same kind of groupings we saw between hot gates and the clusters along that environmental axis that was struggling to put stuff along. So we can kind of get an idea. So we do see a checkerboard pattern where one species is there also means that someone else is kind of like pushing that species out. We find that we do almost kind of see that kind of checkerboard pattern there. And we do kind of notice that, for example, with the hot gates, there's a lot more overlap with the hot gates than there is, for example, with the clusters. So maybe the clusters are doing some of the work, kind of like, pulling stuff apart along that axis, at least in that kind of way. And there's more absences than we would expect on random chance, is why we had that negative. So kind of like wrapping up, we did find, for the hypotheses, just as a reminder, we were saying that if you have a fluvial structure, it's a significant predictor in crayfish assemblage composition. Also that there are distinct clusters of crayfish communities of common profile. And also the last one being about the Clemensian meta-communities being best described as Clemensian. That's it. So the first one, just kind of saying that the fluvial structure is a really good indicator for where stuff is in the basin, and there's good evidence for that. We found that the model that did account for fluvial distances, most species tend to actually have a better or more parsimonious kind of like a model, at least for those, as opposed to the Euclidean model versus the second one just being, kind of talking about, do we have distinct clusters of these communities in PC or in geographic space? We did find that there is at least some support for the idea that there are these distinct communities of common profile across the basin. And the last one, problem child. So for meta-communities, we found that the coherence was significantly negative, but the other two were non-significantly positive. Meaning that, for example, Clemensian view would be that all of them were significantly positive. That would be what we consider a Clemensian view, and we didn't have that at all. So we had what we consider a checkerboard pattern, which is that we had a significantly negative result for community coherence. So it's considered what we non-coherent meta-communities. So kind of wrapping up, some of the main things that we got out of this. So for one, we updated a bunch of the ranges from the species. So a lot of these species haven't really been, either found or kind of like had their ranges updated to Georgia DNR in any of the other databases, sometimes upwards of 40 years. So this work is actually part of a larger work to kind of characterize the distributions of a lot of these species, not just in Georgia, but also in South Carolina, North Carolina. So, and as a result of that, we did also find a couple new locations for Peter's eye. So hopefully, we're trying to see if we can get more of this data into Georgia DNR so we can update its SGN ranking as well. And we did find a bunch of the, we did find three major kind of like predictors for where stuff is in the basin, which are the watershed subbasins or floodgates, the total upstream channel length, which is kind of like a proxy for stream size, and the amount of wetland cover, which can also work, at least in this case, as a proxy for distance to the mouth, or distance from the coast up towards the Piedmont at least. There's there's a lot less wetland cover up in the Piedmont. There's much more as you work your way down to the coast. And I also generated some models, kind of figure out, to kind of explain and predict where we find stuff in the basin. And we found that, for example, the fluvium model actually worked the best for a lot of these species. And we also discovered patterns of community composition that we can actually say we can actually group these into separate categories of these communities where we expect to find, for example, more Peter's eye. We could point to say, okay, the green ones are what we should look for that one. Or if we want to find more little strata, we could point to the yellow dot, say where we would find more of them there. We're the likelihood at least. And so I kind of want to end it there. So do we have any other questions or concerns about the work?
Questions for Reggie? Steve.
So you sampled the two spots in each HUC-10, shows a major, a larger and a smaller. So when you did that, what did you do if water was up on the floodplain? Did you try to stay in the channel or did you go up on the floodplain?
So we did try to go into the adjacent floodplains.
Okay.
That was something I kind of talked about with Checo, with possibly getting boats out there, so we could probably do some more deep sampling, but we didn't end up doing that. And we did, for example, we tried to get as much habitat diversity as we could. So that's why we chose these smaller order and larger order streams. And also some of these, for example, in the HUC-10s that actually had the main stem, that would have been our larger order. But sometimes if you move away from the main stem, it's no longer, for example, that main stem status anymore. So we tried as best we could to make sure we got a decent amount of habitat variation, at least.
Related to kind of that, I think it goes to a question in the chat where somebody wants to know, how did you define how do you measure wetland cover?
Okay, so for wetland cover and also for all the other land cover classifications, we actually got our land cover classification from the NLCD, and I just used ArcGIS to kind of get an idea for using the HUG-12s, which is a step even smaller than HUG-10s, as our kind of like our grain for our study. And we kind of defined how much wetland covers by the number of each of those cells within the greater kind of like buckets that each of those HUG-12s were in. So how many of those were wetland cover versus, there's different classes of wetland cover. So we have woody wetland cover versus kind of like emergent herbaceous wetland cover. So we took each of those, we threw them together in what we call wetland cover class versus, for example, agricultural cover, which was, for example, pasture and cultivated crops. I squeeze those together to form agricultural cover. So I actually had custom classes that I use for each of those groups. So, for example, there's 4 classes of developed cover, so I just smush those all together into one giant developed cover. If that makes sense. Yes. Go ahead, on.
How many crayfish did you go through to get all this data?
So, number of individuals?
Yeah, like a number.
250 to 300.
So you said you found 11 out of the 18
Yes.
species. The 7 that are missing, are these considered rarer species in the Ogeechee? I guess, is there any species that you were surprised that you didn't find?
So, for example, there's quite a few. So there are a couple of burrowing species that we didn't get. And for example, those guys can be harder to sample for. So I try to do my best at, excavating burrows wherever I found them. We did find, for example, 4 of those species that were left out of the model. They were left out because the number of occurrences was less than like a standard, I said 4 is my limit because we had like 37 sites. So they had less than 10% prevalence. And those sites we only found once or twice in all my sampling, unfortunately. So there's a few of those that were very rare, even as out of the ones that I caught. Some of them, we didn't find, mostly just because like sometimes it's the time of year for a lot of these places, they're just harder to find. So I also tried to spread out my sampling across the year to kind of combat this, but I just didn't get lucky. And also, I will say, even if I didn't catch it from my setting, we have found them in other parts of the state of Georgia. So, for example, Lunsey, super common on our Bear Islands, and they're also kind of common as you work your way further south. So, some of these guys, they are there. It just, as part of my study, I just didn't have the luck enough to find them.
Thank you.
So, kind of following up on that question, could part of that be the habitat preference or ecology of some of those species? Sort of like Faxinella, for example, you didn't find. I'm not familiar with the one here, but I know other Faxinella species are more associated with like ephemeral wetlands, vernal pools, ditches, that sort of thing. Could that be part of the reason that you didn't encounter those?
Yes. So I intentionally tried to just do sampling along the fluvial network. So if we do have, for example, species that are only found like in our ditches or wetland habitats. So for example, the Thesis is actually much more common we found in our wetland habitats, and also we found in some of our Bear Islands. But we did find that, for example, that because I had a built-in biasing with my sampling, so only choosing to sample in the fluvial network. There are a lot of our species that are in the basin that are typically only found in some of our ephemeral wetlands, and also some of our what we call lentic or more like the still water sites. So that is actually something that is to be considered. But for the scope of my study, I only kind of focused on the fluvial kind of species that are found. Yes.
A brief note on the prevalence of red swamp crayfish throughout the study.
So as a part of my study, we had not found in the range, but we did have a crew of people from North Carolina come through, and they did find one peacocky in the upper Ogeechee. So as far as we know, it had been found in the basin. Fortunately.
I'm gonna hit you with a 3-part question from Zoom.
Chris, go.
Yeah, not Chris. So colleagues on Zoom want to know, I'm gonna put it as 3 parts. 1, what would you recommend to state agencies for the prolonged conservation of Bear's Peter side there? 2, and maybe related, what would you be most excited to study next, or new questions that you want to ask related to this? And then lastly, what is your favorite memory from the field? So start with state agencies, conservation, preservation of Peter side.
So when it comes to Peter's side specifically, the problem that they were facing was that they don't have information on that species pretty much at all. So part of this work, we've only found, we managed to add another 5 sites that where they are at least, and we're finding like 30 to 50 individuals for the species at each of those sites. So at least we do know, for example, that they do have healthy populations where we do find them. So it's just a matter of finding specifically where they are. So part of this is like this framework for like a pipeline of being able to go from, you know, where we find stuff to making a model and getting these predicted outcomes to formulating like regions of like a common profile for like so we could focus some of our effort in those regions at least. So maybe like not just focusing on the entire basin, we can kind of focus a lot of our resources to those areas. So in this case, focusing on like in those places where in the upper Ogeechee at least, where they were much more likely to find it.
In favorite memory from the field, as we apparently have a class here at 2. Your favorite memory from the field?
So the first time that I actually went out into the field, I remember I was with Nick, and we were just kicking around, and I'll never forget, we actually saw, I think, a little white dog off in the distance. We were just confused, like, what? Shaking our heads, and then we saw the owner peeling through the wood after it. Yeah, that was great.
All right, well, join me in thanking Reggie. You go to the close part of the defense, hopefully celebrating a little later in the day. Thank you all for joining us. Thank you all in Zoom.
Did you appreciate yourself?
I love it. Thank you. Good to see you. How are you doing? Good job.
Close there, so we're right. Yeah, just close that part, stop chair.
Assemble.
Yeah.